Hi, everyone, and welcome to my channel. This is the sixth episode of the Meet the Pro series, a series where I will be interviewing people in the field of biology and genetics to help high school students. In today's episode, I will be interviewing Dr. Cliff Tabin. He is the chair of the genetics department at Harvard Medical School studying evolutionary biology, and I'm delighted to have him with me here today. Thank you for being here, Dr. Tabin.
Pleasure.
Okay, so I would like to begin by asking you to take a trip down memory lane. Could you take a few minutes to describe how you became interested in biology to eventually getting to where you are today?
Sure. So my father was a physicist, and I grew up with him sort of inspiring me to think about the natural world and sort of the wonder of not just how marvelous the world is, but the fact that we can actually understand it. We can pose questions, and initially as a little child, you pose questions and your father answers them, but eventually, or your mother answers them, but eventually you can devise methods of finding the answers yourself, which
Yeah.
that was an extraordinarily fun and exciting thing. And initially, I thought about following my father's footsteps and becoming a physicist myself. That was my plan when I went to college. But the types of questions that were really exciting at the time in physics, and this was a long time ago, was the mid 1970s.
Okay.
The questions that were very exciting at the time were being approached by very large teams working on very large machines that would be able to generate a great deal of energy and be able to have beams of energy that could probe the basic forces of nature. And while those were the questions that interested me, that wasn't the type of science I wanted to do, and that a graduate student at that or a high school student working in a lab at that level wouldn't have an experiment to do, but would be part of a big team.
Yeah.
You would help design a detector at a particular energy level to be able to detect a certain event that would occur, but you wouldn't really be able to pose your own question and model it your own way. So I was looking for something else that might appeal to me, coming with a physical sort of a background. And initially, I thought about biophysics. I thought using physical approaches to understand the structure of matter, a structure of not just matter, but of life, matter that's important for life. So the three-dimensional structure of a protein, for instance, or a nucleic acid, would be a very interesting and exciting thing to think about.
Mhm.
So I went to graduate school at MIT in biology, with that being my initial intent. Right when I went to graduate school, there was a revolution in our ability to probe questions with the invention of recombinant DNA. And for the first time, we were able to isolate individual pieces of DNA, see what they encoded, test their function, and so on. And that opened the door to this whole revolution that's both in our understanding of the world, but also our ability to manipulate it and devise new techniques for helping people and open up enormous doors. So I was drawn very quickly away from biophysics into molecular biology. And then from there, it was a matter of what questions were most exciting to me in biology. As a graduate student, I worked on cancer and the genes that are modified through mutagenesis to give you oncogenes, genes that are involved in forming tumors. And then as a postdoc, I moved into questions that I thought were just more fundamental interest, less medically driven, the questions having to do with how organisms form and how they evolve. And that's where I've been focused ever since. Sort of remembering that I didn't go, today, many people are drawn to biology out of the desire to cure disease.
Yeah.
And it's noble and it's very important. And we have the opportunity to do so much in that arena. The opportunities are almost endless. But when I went into biology, I came, as I said, from a physics curiosity sort of background. I wasn't really thinking about curing anything.
Yeah.
And if you think about the physics questions of the day, the idea that fundamental particles are made up of smaller particles called quarks was a new hypothesis at the time. And testing the quark hypothesis is a part of the standard, it's now the standard model of physics, bringing together the strong force, the weak force. That wasn't about curing disease. That wasn't about helping people. It was about trying to understand in a fundamental way what molecules were made of,
Yeah.
what atoms are made of, what subatomic particles are made of. So in a similar sort of way, what drew me into science in the first place was answering questions. And so that's really where I stayed, and I sort of had a curiosity-driven intellectual path rather than a medically driven intellectual path.
So is your research even now, is that still focused on evolutionary biology?
Well, I would say that the focus is in two areas: evolutionary biology and developmental biology. So if you take a step back, the big questions are, where do babies come from? How do you get from a single cell to a resheet?
Hmm.
It's a pretty remarkable thing. And
Yeah.
it's sort of two, in some sort of, if you want to divide the world up into different categories, two classes of problems there. One is, how do you get all the cell types in your body? How do you get skin cells and nerve cells and bone cells and muscle cells and so on?
Hmm.
And that's a very important question. That has to do with stem cell biology. It has a lot of importance for medicine as well. I tend to ignore that question. The other half of the question is, how do you get the organization? You're not just a mass of random cell types. Why is your arm different from your leg? Why is your thumb different from your little finger? How do you get all of the muscles in your arm attaching the right place to move the bones? How do the different bones get to be the different lengths that they are? When the embryo first forms, your toe bone and your thigh bone are the same size. How come the thigh bone grows so much larger in the embryo? Those
Hmm.
sort of organizational questions are more what's interested me. And the other big question that we focus on is the diversity of life on Earth. How do you get this incredible range of beautiful organisms that make life so rich on Earth? And fundamentally, it's again a question of organization. The difference between a mouse and a cat is not different types of cells. All the cells in a cat are in a mouse and vice versa. The difference is very subtle changes in how you organize the tissue. You again start with a single cell that has the potential to make muscle, bone, tendon, heart, and so on. You have a single cell that can make the same cell types, but as that cell divides and multiplies, the cells are either going to grow into a cat or into a mouse. And so understanding how that happens, it's the same type of question of how you get organization. And those have always been I guess I started out by thinking more about how you make an embryo, how you make a resheet, and how you make many
Yeah.
types of animals. But that largely was because until you know something about how you make one type of animal, you can't think about how you make more types of animals. Once you have a certain amount of knowledge about how you make one, you can think about how you change that program to make different types. So over time, we've sort of branched out. And yeah, so the fundamental focus of my labs remained developmental biology or embryology and evolutionary biology, how the program of evolution how the program of development has evolved to give you the evolution of different forms of life on Earth.
Okay. So I want to kind of ask one question in both, like one with developmental and one with evolutionary. So for
Sure.
development, um, how do our cells, like, how do they know what type of cells they need to become, or like what decides that? And then how do they know how to make the leg that they have to make it look this particular way? Or same with the arm.
So the instructions are in every cell, right? So the DNA is like the blueprint,
Yeah.
the instructions for building your body. Those instructions are the same in your skin and in your brain and in your muscle and in your bone and so on. To get different cell types, it's a matter of which parts of the instruction you're following.
Yeah.
If you have a thick instruction manual, you can go to page 32 and get the
Yeah.
flow chart there and do what it says there, or you can follow the instructions on page 78.
Mm-hmm.
So a large part of the process is opening up certain pages and not other pages. Well, the pages are your chromosomes or your DNA. And there's literally open and closed chromatin, which is a way of saying proteins plus DNA, but the open and closed portions of the chromosomes that can be turned on or off.
Mm-hmm.
And the whole series of events so what you've got to do is you've got to control which pages are open. And some of that is from cells talking to each other. Mm-hmm. A process called embryonic induction. In induction, a cell is going to become a particular thing until it gets a signal. If it doesn't get that signal, it stays in this path, but it gets a signal, it does something different. Okay. Or in other cases, you have cells that go through a series of changes that once you start a process, it's like a cascade. Once it takes the first step, then once the last direction on page 82 says turn the page to page 83, and then you follow the instructions on page 83. Yeah. So part of it is just history of what's happened before. Difference between the arm and the leg, there are proteins called transcription factors, which are proteins that turn on other genes. So a gene makes a protein. So there's a gene that makes a particular transcription factor. That transcription factor will turn on other genes. Mm-hmm. So some of the proteins will be just like building blocks of the cells. Some of the proteins will be signals between cells. What the genes are will vary, but transcription factors are like at the top of the process. They turn on particular sets of genes. So we now know, we can now identify the transcription factor in the embryo that is important for making an arm instead of a leg. Yeah. So in an experimental animal, obviously not humans and not even primates, because you wouldn't want them to do something that would harm an organism like a human, or mess with the human embryos would be very unethical. Yeah. But with a chicken embryo, for instance, you can use tricks to turn on a transcription factor that normally is in the leg into the arm instead, and that chick embryo will start to make an extra leg instead of a wing. You can convert the cells that would have made the arm, would have made the wing, into cells that will instead make a leg by putting in the right one of these transcription factors that basically takes over and says, okay, we're going to turn on the leg genes. Okay. And in some cases, there are specific genes. In other cases, it's just amounts. Because fundamentally, an arm and a leg are pretty darn similar. You would never mistake one for another, but they both have muscle, they both have bone, they both have tendon, they both have cartilage, they both have nerves running through them, they both have blood vessels. Yeah. They're very similar structures. They both have 3 segments, an upper arm, a lower arm, and a hand, or upper leg, a lower leg, and a foot. Mm-hmm. So you're basically tweaking the system. You're saying, okay, well, as you grow out and form a limb, you do this a little bit differently. You make a little bit more muscle here, or you make a little bit longer bone there, and the end result is an arm or a leg. Mm-hmm. And the thing the toggle that says go a little bit more this way than that way are these transcription factors, and one can identify what they are. And so there's sort of 2 levels of understanding here. One is the sort of the broad general level that I'm talking about now. So we now understand that there are such transcription factors that control whether you're going to make an arm or a leg. And then there's the deeper understanding of, okay, well, exactly what is that transcription factor? Exactly what is it doing? What genes specifically is it turning on, and how does it turn them on? What do those genes do? And getting down to the details and the weeds of the instructions.
So it's like it's a network of transcription factors plus minor tweaks depending on what you're making, so that you get your arm, you get your leg, you get the head, the feet, I guess the brain, or it's just about the minor tweaks.
Yeah, and it's just, and again, there's a process. Embryogenesis unfolds. You start with a single cell, you end up with the whole body. Mm-hmm. So before you make a brain, you make a neural tube that's going to be what's going to ultimately become your whole nervous system, from your brain through your spinal cord. Then the different sets of genes tell one part of the tube you're going to become spinal cord, another part you're going to become brain. Then other genes say you're not going to be just brain, you're going to become the forebrain, you're going to be the hindbrain. Mm-hmm. And then within that region, different transcription factors will run different cells within that region saying, okay, you're going to be this type of neuron. You're going to be that type of neuron. You're not going to be a neuron at all. You're going to be a glial cell, which supports the neurons. So there's a whole process. And at each step, it's carefully controlled, both by signals from cells talking to each other. The one cell becomes a particular type of neuron. It tells the cells around it, don't make the same type of neuron as I am, do something else because I'm already here. Or I'm here and I need this other type of cell to help me, but go become that.
Mm-hmm. Mm-hmm.
And when I say talking to each other, obviously they're not talking verbally.
Yeah.
They're sending out these secrete molecules that are received by other cells that influence through a series of biochemical changes what happens within the next cell.
Mm-hmm. Okay. And this is an evolutionary question. I kind of got this from reading through some articles on your website. So, how can cavefish survive or even thrive in caves where resources and light are both limited?
Okay, so we work on a large number of different topics in my lab.
Mm-hmm.
One of them is the evolution of cavefish.
Yeah.
So it's an interesting system to think about from an evolutionary standpoint, because you have a species of fish that got trapped in a cave. And as you said, over evolutionary time, they evolved to be able to survive and thrive there.
Yeah.
In a place that's totally different from where they used to be. When you think about global warming, you have the environment changing over time. Usually one thinks of environmental change as being very slow and gradual. And we're unfortunately accelerating it to an enormous degree with global warming and climate change, which makes it very hard to adapt for animals to keep up quick enough. Mm-hmm. But with cavefish, you're talking about something that happened abruptly in a single day. A fish is swimming around in a river, perfectly happy with its little environment, got to hide from its predators, but it's got plenty of food, lots of things it recognizes, plants and other things. All of a sudden, it gets washed into a cave, down a pipe, and now it's stuck, and it's totally dark. The bad news, well, the good news is no one wants to eat you because there are no predators down there.
Yeah.
Bad news is there's nothing for you to eat.
Yeah.
It's totally dark. And when I say there's nothing to eat, there's this whole web of life where light is absorbed by plants through chlorophyll, which allows the plants to have energy. The plants grow, plants get eaten by small animals, bigger animals eat the small animals, bigger animals eat the mid-sized animals, and so on. Well, there's no light in the caves, there's no photosynthesis, the whole chain is gone. Right? So without plants, you don't have small animals, you don't have bacteria, you don't have anything. Mm-hmm. So you do have occasionally sort of decaying matter washing into the caves. Okay. Some caves have bats in them, so you have bat droppings. So but it's nothing like the richness of the river that they came from. Yeah. So, and important to note that when there are floods into the caves, lots of animals get washed into the caves. Mm-hmm. Of all the animals that get washed in the caves, in the particular caves we're in, only one vertebrate, only one type of fish survives there. And it's happened in many, many caves throughout this region of Mexico. And it's always the same type of fish. Okay. So there's something about these fish that allow them to get through that bottleneck of where the hell am I, how am I going to survive, and find a way to evolve to be able to make it there. Yeah. And all the other types of fish that get washed in there just starve to death. So what do they have to do? What do they have to do to survive? Number one, they have to find their way around. Mm-hmm. So they've evolved. They can't see anything. So the sense of sight is not useful. And in fact, they lose their eyes entirely. They evolve to be blind because eyes are very energetic. It takes a lot of energy to keep an eye functioning. And if you're going to be in a cave with little food around, you don't want to waste energy on something that's not doing you any good. So they get rid of the eyes. Mm-hmm. But they have to find their way around. So they take other sensory systems and expand them. Like the fish in the water, in the river, their ancestor, just like you and me, have taste buds in their mouths for tasting food. These fish have taste buds over their whole head. So they're basically tasting with their cheeks and their eyebrows and everything else,
Okay.
which makes them really sensitive for finding food.
Mm-hmm.
And that's true of multiple sensory systems. Their lateral line, which senses water pressure, is greatly expanded. So they heighten other sensory systems. And they get by in eating the small amounts of debris that comes in, but they also they're faced with a situation where debris gets washed in the floods. The floods come in the rainy season, in the summer, and the spring. All winter, when it's dry, there's no floods. They have to get through this basically half the year. They're starving every year.
Yeah.
So this is a fish that has to evolve an ability to not lose weight rapidly when there's no food there.
Mm-hmm.
And when there is food there, they have to put on weight really rapidly.
Yeah.
So they can't waste a single molecule of sugar. Anything that gets into the water that they can eat, they have to pack it on as fat and allow themselves to store lots of energy. Mm-hmm. While the river fish eat year round and basically, like us, can remain healthy and fit by eating a little bit each day, the right amount to balance their calorie needs.
Yeah.
These fish have to go through feast and famine all the time. So they have to totally change their metabolism. And they do it in some very curious, interesting ways. One of the things they do is they dysregulate sugar metabolism. So effectively, they have many characteristics of a diabetic, but they're not diabetic. Diabetes is a disease.
Yeah.
Terrible condition. These guys can tolerate very high blood glucose, so that when there's sugar around, they can just pack it into their blood, absorb everything. Okay. So another thing that they do is they get when there's food around, they get very obese. They eat huge amounts of, when there's a flood and it brings in some debris that's decaying, they eat everything they can. They get to be quite fat. Mm-hmm. Now, if you get very fat, it's bad for your health. You know that. We have an obesity epidemic. We call it an epidemic because it's a bad thing.
Yeah.
It's not just bad for humans, it's bad for most animals, but not all.
Not
you also cavefish, or hibernating bears.
Yeah.
So they have to change their metabolism and their physiology so that being very fat doesn't bother them. In fact, very obese people get something called fatty liver disease, where the liver gets very fat and it destroys the liver. It's a terrible disease.
Okay.
Cavefish store fat in their liver. They store fat everywhere. They don't care. So now, we're interested in how this happens. So one of the great things about this particular species for study is that you can breed this cavefish and the river fish cousin that still lives the way the ancestors did, and they're fertile. So you can look at the offspring and say, if you breed a cavefish with a river fish, what do its children look like? If you breed two of the children together, what do they look like? Yeah. And you can watch and see some of the fish will inherit more cave traits, some will inherit more river fish traits. And by following which genes go with which traits, you can track down the genes that are responsible for it and understand how it evolved. And one of the things that comes out of that is that there are genes that are protective. So yes, there are genes that say get really fat as quickly as you can, but there are other genes that say these genes decrease the influence. So one of the problems with being fat for a human is fat causes a lot of inflammation. Your immune system starts hurting you by having systemic inflammation. So the cavefish turn on genes, along with when they get fat, they turn on other genes that tell them, that say, don't get hurt by this by turning down the inflammation. It's sort of like an anti-inflammatory response. Well, the reason we're interested in the problem is because I just want to understand how these fish learn to adapt to this weird environment. But although it's not our reason, this type of research will lead us to the realization that there are these protective genes, and maybe understanding something about how the cavefish protect themselves from being fat will give us some treatments for human obesity. So although we do very basic research, multiple times through the course of my career, we found things that have been things that really could help people in some ways. And although that's not the direction my lab takes it, it gives other researchers who are interested in obesity, for instance, clues that they can follow up. So if this gene is protecting a cavefish, maybe it'll help protect my patients. And then a doctor who works on obesity can take that and run with that. So, basic research is a little bit different from applied research. So with applied research, you study exactly the problem that you're interested in. You're interested in cancer, you study cancer. Basic research, you study very fundamental processes, following very just curiosity and your interests. But you never know when it's going to lead to something that's going to be important. So, for example, a colleague of mine who used to be in our department, Jack Shostak, won the Nobel Prize for discovering an enzyme called telomerase, which is involved in making the ends of chromosomes protected. So your chromosomes would get chewed back over time if they didn't have these protective ends. And the ends are also very important for replicating the chromosomes. Well, it turns out with aging, chromosomes get shorter, the ends get shorter and shorter and less effective, and it's part of the problem of aging. But cancers, which basically live forever, and that's one of the problems with them. In tumors, they very often activate this enzyme that extends the ends of chromosomes, thereby keeping them sort of in an immortal state by turning on this gene telomerase. So by discovering telomerase gave rise to new directions for therapies against cancer. How did Jack find this cancer, make this big cancer discovery? Was he studying human tumor cells? Was he studying tumors in mice? No, he was studying yeast. And he was looking at a yeast and trying to understand how a yeast chromosome replicates. And he found that there's this particular sequence at the end of the chromosome in yeast that's necessary for the chromosome yeast for the yeast to replicate its chromosome. And a colleague of his, Liz Blackburn, whom he met at a conference, had done the same thing in another totally different organism called Tetrahymena. They isolated the end of the chromosome and found it had a unique DNA sequence. But the sequence at the end of the Tetrahymena was different from the sequence at the end of the yeast. So Jack said, I wonder if the Tetrahymena end will work in a yeast. And they did the swap and showed that it worked perfectly well in the yeast. He's just trying to understand ends of chromosomes. He wasn't thinking about cancer. He wasn't thinking about aging. But from that experiment led to the discovery that what the specialized ends of the chromosomes were built by was this unique enzyme that made the ends unique, called telomerase. And so by trying to understand ends of chromosomes in yeast and Tetrahymena, Jack found an enzyme which nobody would have looked for this enzyme if they were studying cancer. You wouldn't have gotten there by studying cancer. People studying cancer weren't thinking about ends of chromosomes. But once you found this, all of a sudden it's wow, this gene gets activated in tumor cells. Maybe it's really important for tumors to have this enzyme going. Maybe if we can block this enzyme, it'll be a way of countering tumors. So fundamental research leads to things that you wouldn't get to otherwise. So I think fundamental research is important, but so is applied research. You need people who take it there and take the discoveries that labs like mine make and turn them into things that help people.
Okay. I want to end with one last question. So AI has really been taking over a lot of molecular biology, especially with respect to protein structures. Can we use AI to study evolution?
Well, there are sort of two levels where I think that AI could be brought into play. One is in modeling evolution, and you can think of how you could use AI as a way of understanding an entire process of evolution. The other, just by having a computer version of it, not real animals, but see what it takes to evolve things in a computer. But the other thing that AI does is allows you to look for patterns. You can train in machine learning, you can train the computer to recognize specific things, and you can say, okay, are there things, we talked about the cavefish before, are there things about the cavefish that are different from the river fish that we hadn't noticed before? when you have offspring when you breed a cavefish and a river fish, what sorts of things are different between the two? We can say, well, here's a list of genes that are different, here's a list of genes that are always inherited along with a particular cave trait. Let's say ability to tolerate high amounts of fat. Which of those genes are important? Well, AI can look through those genes in a lot faster way and a lot deeper way than humans can, to try and zero in on which of the many genes that are changed are the ones that might be most significant. So I think AI can be an important tool. Of course, at the very earliest stages of AI,
Yeah.
AI is a young field, and I think you're going to see more and more applications of it in biological sciences. It's not something we use particularly in my laboratory.
Yeah.
Yet.
Okay. And finally, I want to end with a sort of fun question. If you could choose any extinct species to bring back, which one would it be and why? And you cannot say dinosaurs.
I'd say near humans. I'd say it'd be really interesting to see what the difference, how different from, this is a thought experiment. This isn't
Yeah.
something we would do ethically,
Yeah.
but how different from modern humans were Neanderthals, for instance?
Okay.
I mean, were they basically humans who looked a little bit different? I mean, they're very close relatives.
Mm-hmm.
Or are they really do they have different capabilities, different cognitive, how do they view the world?
Yeah.
Do they see the world the same way that we do? Do they have the way they would approach a problem be the same way I'd approach a problem? How about if you go back further? So, I mean, how much of the difference between an Australopithecine and a modern human is cultural differences? They just never had the opportunity to learn that it took millions of years to accumulate our knowledge. How much of it is that we actually have a larger brain than an Australopithecine?
Mm-hmm.
Be very interested to see how they behave relative to us and relative to chimps. If I were to pick an animal, that's what I would pick.
Okay. That's quite unique, actually. Everyone that I've asked before, they've always said like some sort of animal. Like I've gotten megalodon, I've gotten the thylacine. I've just gotten animals before, but your answer was very unique, so that was nice. Um, but yeah, I think for today that's all we have time for. So thank you, Dr. Tabin, for the interview.
My pleasure. Thanks for asking me.
Hmm. Bye bye.